There is an animal alive on this planet that sees a universe you will never see. Not metaphorically, not in the way we say a dog smells a different world. Literally, physically, it perceives colors that do not have names in any human language because no human has ever seen them. Its eyes contain 16 types of color receptors. Yours contain 3. You are, in the most precise scientific sense of the word, functionally blind compared to this animal. But color is the least interesting thing about it. The mantis shrimp is not a shrimp. It is not a mantis. It is a stomatopod, a lineage of crustacean so ancient and so biologically separate from everything else on Earth that scientists have spent decades arguing about where exactly to place it on the tree of life. It has been evolving independently for roughly 400 million years. To put that in perspective, when the mantis shrimp's ancestors first appeared, there were no trees on land, no reptiles, no dinosaurs. The Atlantic Ocean did not exist. This animal has been refining itself since before most of the world we recognize was assembled. There are more than 500 known species of mantis shrimp, ranging from a few centimeters to over 30 centimeters in length. They live in tropical and subtropical waters, mostly in the Indian and Pacific Oceans, hiding in burrows and rock crevices. At a glance, some look almost delicate, translucent, almost jewel-like. That appearance is one of the most successful deceptions in the animal kingdom. Marine biologists who work with them have a habit of calling them by a different name: thumb splitters. Not because they are aggressive toward humans by nature, because if you are careless, if you reach into a tank or a reef crevice and the mantis shrimp decides you are a threat, it will split your thumb open before your nervous system has time to process that anything happened. The strike is that fast. We will come back to exactly how fast. There are 2 types of mantis shrimp, and they have solved the problem of killing in 2 completely different ways: the spearers and the smashers. Spearers use barbed appendages to impale soft-bodied prey, fish, worms, shrimp, with surgical precision. They are ambush predators, patient and explosive. Smashers are something else entirely. And it is the smashers that broke physics. In the 1990s, a scientist named Sheila Patek pointed a high-speed camera at a mantis shrimp. What she captured changed what we thought was physically possible in the biological world. The peacock mantis shrimp, one of the largest smasher species, strikes its prey with a specialized club-shaped appendage called a dactyl club. The strike travels at roughly 23 meters per second. That is the acceleration of a bullet leaving a .22 caliber rifle from a creature the size of your hand. But acceleration alone does not explain what happens next. When the dactyl club strikes, it creates something called a cavitation bubble. The movement is so violent, so sudden, that the water around the club vaporizes on impact. A bubble of near vacuum forms and then immediately collapses. That collapse releases a second shockwave, a second strike from nothing. For a fraction of a second, the temperature inside that collapsing bubble reaches approximately 4,000 degrees Celsius. That is roughly the temperature of the surface of the sun in your aquarium, next to the coral. The peacock mantis shrimp can strike with a force roughly 1,500 times its own body weight. It has shattered aquarium glass. It has broken the fingers of marine biologists who handled it carelessly. One specimen in a laboratory in California spent 3 weeks methodically cracking its way through a supposedly shrimp-proof tank before escaping into a drain. Scientists nicknamed it the thumb splitter. They rebuilt the tank. It cracked the new one, too.
Let us return to the eyes, because the punch is the part people talk about, but the eyes might be stranger. The human eye has 3 types of photoreceptor, cones sensitive to red, green, and blue light. From those 3 channels, your brain constructs every color you have ever seen, every sunset, every painting, every face.
The mantis shrimp has 16. For decades, scientists assumed this meant it experienced a richer world than us, an explosion of color beyond imagination. Then they tested it. The results were deeply unexpected. In controlled experiments, mantis shrimp were actually worse than humans at distinguishing between similar colors. Not better, worse. The 16 channels are not used for color discrimination the way ours are. They appear to work more like a barcode scanner than a palette.
Instead of mixing signals to identify a color, the mantis shrimp's visual system checks each channel independently, a binary yes or no across 16 different wavelengths, and computes the result almost instantly. It does not experience color the way you do. It identifies it, faster, more efficiently, with less cognitive load. It is not seeing more. It is seeing differently in ways we still cannot fully comprehend.
And the eyes move independently. Each eye can rotate on its own axis, pivot up, down, and sideways, independently tracking 2 completely separate objects at the same time. The visual cortex of most animals processes one unified field of view. The mantis shrimp processes multiple. Neurologically, it is unclear how it integrates all of this information. Some researchers have suggested the eyes themselves do part of the processing, that cognition happens before the signal ever reaches the brain. The implications of that idea have not been fully explored.
The dactyl club, the striking appendage, has been the subject of intense study by materials engineers, aerospace designers, and military researchers, because what they found inside it should not be possible. The club is built in layers. The outermost surface is a hydroxyapatite ceramic, the same material as human bone, but organized at the nanoscale into a helicoidal pattern. Each microscopic layer rotates slightly relative to the one beneath it, like a spiral staircase. This structure dissipates crack energy. When the club strikes rock, crab shell, or glass, the impact force is not absorbed by one point. It is rerouted, redistributed, spiraled outward through the helicoidal matrix until it becomes harmless vibration. Engineers at UC Riverside and MIT studied this structure and have been attempting to replicate it in carbon fiber, aircraft panels, and body armor. They have not yet matched it. A creature that has been fine-tuning this structure for 400 million years is, as of today, still ahead of our best materials science. Now we need to talk about what the mantis shrimp does to other animals. Because the punch, the eyes, the architecture, all of that is the animal at rest. This is the animal at war. The peacock mantis shrimp primarily hunts crabs, clams, snails, and oysters, animals that evolution spent millions of years armoring. The mantis shrimp does not care. A crab's shell is calcium carbonate, the same material as marble. The mantis shrimp will strike it at the precise structural weak point, not randomly, but at the exact location where the shell is thinnest, 3 to 5 times in rapid succession. The crab is not stunned. It does not have time to be stunned. The shell simply opens.
But the moment that genuinely unsettled researchers, the observation that has never been fully explained, is what happens after the kill. Mantis shrimp, in multiple documented cases, have been observed striking prey they are not going to eat, striking it repeatedly, leaving it. This is not feeding behavior. This is not territory defense. The closest behavioral analog in the scientific literature is practice. There is a word researchers have been reluctant to use because it carries implications they are not ready to fully commit to. The word is play. The mantis shrimp's brain is not large. By conventional neuroscience, it should not have the cognitive architecture for play. And yet, in captivity, the mantis shrimp has been observed doing something even harder to explain. It learns the faces of its caretakers, individual human faces. Across multiple studies, captive mantis shrimp have shown consistently different behavioral responses to familiar humans versus strangers, not based on smell, not based on sound, but based on visual recognition of specific individuals. There is a word for this in the animal cognition literature. It is a word we typically reserve for mammals and birds. The word is recognition. The mantis shrimp can tell you apart from a stranger, using eyes that process reality in a way no human brain has ever experienced. From inside a tank, it is technically capable of breaking out of. We have been studying this animal for over a century. We still cannot fully explain its eyes. We still cannot replicate its armor. We still cannot say with confidence whether the thing it does after a kill, when there is no food, no threat, no survival purpose, is something we should be calling by a more uncomfortable name. For most of human history, the ocean was a place we crossed, not a place we studied. The reef was something sailors avoided. The creatures on it were, at best, curiosities. The mantis shrimp was discovered, formally described, cataloged, and then largely ignored. It was too small to eat, too aggressive to keep in tanks with other animals, too strange to fit into existing categories. It took a materials engineer, not a biologist, not a zoologist, pointing a slow-motion camera at one in 1998, looking for something entirely unrelated, to make us realize what was living on the reef this whole time. The Dactyl Club is now informing the design of aircraft panels and military helmets. The visual system is influencing the design of cancer detection cameras, devices that read polarized light signatures in tumors that are invisible to human eyes. The spring mechanism has been studied by robotics engineers building prosthetic limbs. A creature we ignored for most of recorded history is now teaching us how to build things. There are approximately 500 known species of mantis shrimp. New species are still being discovered every year, and they have been here, evolving, improving, perfecting for 400 million years.
long before we arrived, long before we were even possible. Which raises a question we have barely started asking. If the animal we ignored for centuries turned out to contain answers to problems we were struggling to solve, what else are we ignoring? The reef is not just a habitat, it is the oldest laboratory on Earth, and most of its experiments are still running.